All right, hello everyone. We're the Turing Sour Snakes, and our project is called Biological Connectivity Patterns as a blueprint for efficient neural architectures in reinforcement learning, which is a bit of a mouthful, but I will go into it. Nima, can you give me the next slide? Oh, okay, so our main idea is that we basically want better artificial neural networks. We want them to be more efficient, more computationally efficient, more interpretable, and they should learn much faster than they currently do. So we can continue this line of just pure ML research, and that will probably get to that point eventually, or we can look for a shortcut or a guide from biology. And organisms have been evolved to be very, very efficient in whatever environment that they're set up in. And so, depending on what environment that we want to have a neural network perform in, we can look to various connectomes and take a inspiration from those. So what we're doing is based off C. elegans, and we're just using that because it's a maximally simple, fully mapped connectome. And we have some RL swimming agents, which we'll go into. But this general idea, Nima, next slide, please. This general idea goes far beyond just a very simple worm. So this is a perfect example where a giraffe can learn to walk just minutes after it's been born because it's very, very highly specialized for this environment. So the end goal of our research is to get to these more complex model organisms along this entire research agenda. But for now, we're studying the C. elegans. So we can get far more efficient than like a totally tabula rasa blank slate RL policy network. Nima, next slide, please. So we can't just say that these connectomes are more efficient. We have to quantify those as well. So how we're doing this is through scaling laws and reinforcement learning. And what these do is they just measure how much better a model gets and how it performs when you give it more compute or parameters or training data. And people have found that larger models tend to just generally do better in all regards, but that comes at a high cost in compute. And so what we're going to show a bit later on is that C. elegans models, which we're calling neural circuit architectural priors or NCAP, if we can model it based off of that, then it does far better and has similar levels of performance at far fewer parameters. So Ruman's going to talk about the methods here.
Right. So we had a simple reinforcement learning setup where we had our Mojoko environment, which is a physics-based physics simulation environment. There, our agent is a swimmer agent. It's a swimmer worm, simulated swimmer worm. Well, and the task is rather quite simple, which is just to swim at a certain speed and stability. Um, in our setup, which is a reinforcement learning setup, where an agent learns by taking decisions and getting feedback in the form of a reward, and based on that, its policies are updated. There are various ways to update that policy. We opted for PPO, which is an active critic based system, whereas where it incrementally update the policy and have a good balance of exploration and exploitation. Exploration as in it will explore new things, exploiting as in it will do things that it has learned works in the past training grounds. Um, our reinforcement learning algorithms are notoriously difficult to implement. So we went with stable baseline 3 library as our choice of implementation. It allowed us to perform experiments at a much faster rate. Even next slide. So, going a little bit into the NCAP architecture, we took it from, it is from the research that we have cited. You can see the citation on the lower left side of the slide. Um, the lower right side shows you the neural circuit of a nematode C. elegans worm, which has these muscles which does swimming through this wave-like propagating way, and it has these muscles which are responsible for proprioception and activating and inhibiting the certain muscles on either side uh, of the nerve. Using a combination of B and D neuron, it has specific neurons, AVD and SMB for controlling steering and speed. So the researchers took this biological circuits and created this computational neural architecture for which directly mirrors this. It has these same neural models and oscillators, which mimics this wave-like propagation motion throughout the system. So the NCAP model is quite different from a multi-layer perceptron MLP in the sense that it is a sparsely and much more structured connected architecture. And the research showed that it is able to train with much fewer parameters and is much more efficient. In the next slide. So we went and validated the research on which our work is based on. And as you can see this current from the video on the right, that this swimmer agent is able to swim with good speed and stability, which is using the NCAP architecture. Paolo will discuss how we stacked it up against NLP.
All right. Yeah, so we try to compare with MLPs or multi-layer perceptron. We have several like design choices to make. And here I've plotted some, for example. So one axis of manipulation for the architecture is how wide we want our nectar to be. And so, of course, like by increasing the width of the nectar, we will increase the number of parameters. So we will trade the capacity for expressing complex behavior with the efficiency, because we need to train more parameters. And of course, the input and output will be fixed by the dimensionality of the problem. In this case, the number of joints and will be our actuator, so the output, and the inputs will be the observation from the environment, so the state of the joint, the speed, and so on. But then another axis that we can try to tune and explore is the depth, for example. So in principle, we don't know whether either narrow or a deep network works best for this particular behavior, or again, we have complete freedom in this sort of artificial system. So, for example, we can explore something more exotic. So, have like say a very wide layer followed by a very shallow one, and the depth can change and so on. So, Nima, if you go to the next slide now, the whole idea was basically, okay, let's take a completely zero prior approach and just try to explore the space of possible architectures that are out there. So what we do is basically have like an efficient implementation, and we run several simulations, around 200 training simulations on a remote cluster, and in where we systematically explore and generate random variation of the MLP architecture by spanning across the axis of depth and the axis of width. And of course, these two axes have separately increase the number of parameters. So as you will see, we will move from architectures that are in generally very narrow and shallow, which will have a very low parameter count. In this sense, they are very efficient in terms of number of parameters. And the whole question is, can we learn good behaviors with these very small networks? And on the other hand, as we go towards the upper right portion of this plot, we will explore more wide and deep networks. Of course, this architecture will be more capable, but we will be trading this capacity by actually needed to train more parameters, so we will lose in terms of efficiency. So the whole idea is basically try to train a lot of architecture and see where do we land in terms of like accuracy of our output behavior, and then we will compare this with the biological counterpart, which is represented by the NCAP. So Nima, if you go to the next slide. So width and depth are not the only two factors that we can manipulate. So for example, if we fix the width and the depth of a network, we will still have this other parameter that we can try to play with, which is the sparsity. Because one can reason that, okay, maybe I need a sufficient width or sufficient depth to actually come up with an interesting behavior, but then I don't require the full extent of the connectivity matrix. So I can just pair some connection. And here in red, we have highlighted the ones where we just randomly erased this connection. And maybe by this way, we will still be capable of having a fairly large and deep network, but we are reducing this way the number of parameters. So we are trying to be more efficient. So achieving efficiency by increasing the sparsity of our parameters. And this network will be represented by this black square in the plots that Nima will show. So if we go now to the next slide, we came up with another final attempt at trying to still improve our efficiency, and we leverage the so-called teacher distillation process. So the whole idea is that during reinforcement learning, you need to learn in an environment from experience, and this is in general very difficult because the information is quite sparse. So you need to actually reason, okay, I got a good reward, so I need to update my parameters, but you need to act and so on. So there are trade-offs in terms of exploring and exploiting. So the whole idea of this distillation process is that you let a fairly big, so wide and deep network do the heavy liftings, so learning a behavior, and then you take a second network, this is called the student network, which is way smaller in terms of number of parameters, and you just try to copy the learned behavior. This is way easier because you don't need to explore and come up with a solution on your own. You just need to implement it and copy. And so we tried it as another way to actually try to boost the performance and the efficiency of our MLP architecture. And in the plots, this will be shown as these gray circles. And so now I will let Nima explain the results in detail.
Okay, so in terms of results, let's just start with the one layer MLPs. So all the panels that I'm going to show you on the x-axis, we're going to have the number of parameters, which is coming from the number of neurons and weights. And also on the vertical axis, we will have the best average return, which you can assume is going to be the measure of performance for the networks. Now for the one layer MLPs that were trained and then we did that like sparsity weight reduction too. The first observation that we can make here is that we see lots of models with low number of parameters that are outperforming the ones with higher number of parameters. So the contrast between top left and the bottom right here. If you also look into the networks that were trained using distillation, so as Paolo said, these are the student network that learn the knowledge from the fully functional teacher networks. You can also see the same pattern. So there are still lots of networks that are outperforming more complex networks. The other thing that we can do is to fit the sigmoid function that was previously shown by Austin. They kind of show how knowledge could be learned and preserved in paradigms as a function of number of parameters. And you can see that there is the saturation point, which after there is no more learning happening as a function of number of parameters. And another interesting observation is that all these networks have more than 100 parameters and they cannot achieve what NCAP can do. So NCAP has only 4 parameters and it's outperforming all the other networks that are below this dashed line here. Which is all these observations are suggesting that connectivity matters a lot. Now, if you look into a deeper network, so if we extend the depth to 4 layers, you can actually see the results are similar. You also can see increasing depth means increasing more number of parameters unnecessarily because they're not doing so well. And there's this shift on the intercept of our sigmoid functions, which is showing like, hey, the optimal learning is happening much later as you increase the depth of your network. So yeah, essentially we're saying choosing the right connectivity for your specific question is really critical here. Now I hand it over to Zahra for our conclusions.
Yeah, okay. So to conclude, our work highlights that biological neural networks can serve as a valuable blueprint developing more efficient AI architectures. In other words, yeah, we showed that scaling alone is not a key, and connectivity seems to have a very crucial role here. And this conclusion is based on our twofold findings. On one hand, we observe that NCAP as a biospire architecture occupies a highly favorable position in the performance to parameter trade-off. And on the other hand, we saw that many small yet well-connected MLPs actually outperform larger MLPs with suboptimal connectivity. And looking ahead, there are several exciting directions to explore. First, we aim to investigate additional biological architectures to better understand how they fit within this framework, and also to see if they follow any specific interesting scaling position. And second, as we move towards more brain-like architecture, a key question arises. So what truly defines brain-likeness? And to explore this question, actually we turn to methods like RSA as a potential tool to quantify how closely our models can get and align with neural representations. Okay, so thank you very much for your attention, and we will be glad to answer your questions.
Great